go. The topic for my presentation today is related to using Raman spectroscopy coupled with machine learning to evaluate some food chemical and microbiological hazards. So firstly, I want to introduce some basic fundamental issues about Raman spectroscopy. So maybe some of you heard about vibrational spectroscopy before, and you heard about infrared spectroscopy before. So both infrared and Raman belong to vibrational spectroscopy. As you may know, molecules have different vibrational modes in the environment. So for example, stretching, wagging, etc. And there will be some interaction between the laser introduced from the equipment to the interaction of the molecules. So this interaction will generate a different type of the spectroscopy model. So for the IR, this is we call the absorption. And for Raman, this is we call the scattering. So that's the major difference between infrared and the Raman, because IR is for absorption and the Raman is for scattering. So anyway, eventually, due to this interaction between the incident light, the laser, and the vibrational mode of the molecules, it eventually will generate a spectra. So every chemical or every compound has the specific composition, and each functional group have a specific vibrational mode. Eventually, each chemical, each compound will generate a specific spectral fingerprinting feature. So this is very similar to each human of us. We have a unique fingerprint. So on the basis of this, we can do a lot of detection, discrimination, and identification of any unknown or known compounds, or even large molecules like bacteria and viruses. So this is a confocal Raman spectroscopy system that we performed before. And the laser, as you can see here, is a green laser. The wavelength is 532 nanometer. So this gives you a general idea that you need to work in a dark room for all the Raman spectral collection. So this is about 10 years ago since we started to work on using Raman for a lot of different microbiology study. This gives you a general idea how you can use a confocal Raman system for the characterization of the bacteria. So for this paper that we published about 10 years ago in Journal of Clinical Microbiology, we used the Raman spectroscopy coupled with chemometrics and also multivariate statistical analysis for the speciation and the detection of a foodborne bacteria that we call the Campylobacter. So there are a lot of species of Campylobacter, for example, Campylobacter jejuni, Campylobacter coli, Campylobacter leri, etc., etc. So what we do is we got the sample. This could be either the sample from the environment, from the agri-food, or even from the human clinical samples. We firstly do the enrichment, selective enrichment to get the Campylobacter culture, and then we do the incubation and get the colony on the blood agar. So after we do the collection of the colony from the agar plate and transfer into the this is we call the gold slide, microarray slide, and then we deposit each individual Campylobacter sample on the surface of this gold-coated microarray slide. We put the slide under the microscope, and then we introduce the laser to shoot or illuminate on the surface of each individual bacteria cells. So the laser that we use for this paper is about 532 nanometer, and according to the schematic illustration, you can see that all the relay scattering signals will be blocked by the beam splitter, and only the Raman scattering signals will pass through the filter and eventually be magnified and shoot onto the CCD detector to generate such a Raman spectra. And because each individual species of the Campylobacter bacteria have a different chemical composition, so each species of the bacteria will have a different Raman scattering signal. So on the basis of the unique Raman fingerprinting spectral feature, we can do the detection and the speciation of different species of Campylobacter bacteria. So the major advantage for this method to do bacteria speciation is the speed. So we can finish the speciation of the bacteria once we have the pure culture just in a few seconds. So this is very fast compared to the conventional typing methods. A major challenge or a major part of the work is to do data analysis after we receive the Raman spectra. So in the lab, we have a lot of different ways to handle the high-dimensional Raman spectra. So this include both the unsupervised chemometric models and also the supervised chemometric models. So many of you may heard about the principal component analysis. So this is an unsupervised cluster analysis. And some of you may also heard about the partial least square regression model. So this is a representative supervised chemometric models. But during our investigation in this field for many years, we figured out that no matter chemometric or no matter unsupervised or supervised chemometric models, there are a lot of limitations. That's why we start to step into the machine learning algorithm and try to use the machine learning to offer a better and more precise data analysis on the basis of the Raman spectra that we collected. So in the lab, we use MATLAB, we use the R and also the Python to write the coding and the programming. And on the basis of those chemometric models, along with the machine learning models, we can get a better understanding about the Raman spectra that we received. So this is an example that we published a paper recently in Applied Environmental Microbiology. We used the Raman spectroscopy coupled with the machine learning algorithm, especially this is called convolutionary neural network, to accurately specify different species of another emerging foodborne bacteria that we call the Archobacter. So I don't want to go to details regarding this big table, but what I want to demonstrate is the overall accuracy by using the Raman spectroscopy coupled with the machine learning algorithm can achieve the overall accuracy over 94%. So this is a very impressive result compared to the result that we ran before using the Raman coupled with the conventional chemometric methods. And back to another research field that we use the Raman to study the antimicrobial effect. So about 10 years ago, we identified a lot of organosulfur compounds from the garlic. And we see those organosulfur compounds from the garlic has a very significant antimicrobial effect. And we want to study the mechanism, how those volatile organosulfur compounds can inactivate the pathogenic bacteria. So we used the Raman and infrared spectroscopy for the first time to identify that those garlic-derived organosulfur compounds can freely penetrate through the bacterial cell membrane and the cell wall, and then combine with the cell-containing enzyme inside of the different pathogenic bacteria, including the E. coli, Salmonella, and the Listeria. And that's the reason how those sulfur compounds can inactivate the foodborne pathogenic bacteria. And we've successfully used the Raman to monitor the whole dynamics, how the sulfur-containing compounds penetrate through the bacterial cell membrane and combine with the cell-containing enzyme inside of the garlic. And this series of the works receive a lot of media attention, and we got a lot of interview to talk about how the garlic compounds can significantly inactivate the pathogenic bacteria compared to the conventional antibiotics that we used. So this is a recent example that is an ongoing study in my lab at McGill right now. So we built this homemade Raman optical tweezer. So what is optical tweezer by using the Raman spectroscopy? So for the conventional Raman spectroscopy, you can only collect the spectra on the basis of the biological samples or chemical samples you put onto a substrate. However, it's very difficult to handle the live bacteria or the live microorganisms in an aqueous environment. So for the optical tweezer, there are two purposes of the laser. So the first purpose of the laser is to shoot onto the biological sample, and then you can collect the Raman spectral features. And the second purpose, which is more important, is the optical tweezer will generate an optical trap. So this trap can kind of capture the bacteria or the viruses and manipulate the movement of the biological samples within a microenvironment, such as a microfluidic lab on a chip. So in this product, we used the Raman optical tweezer coupled with the microfluidic lab on a chip to successfully capture detect and discriminate single bacterial cell. And after we got the ID of the single bacterial cell, we can artificially manipulate how the bacteria will move inside of different chambers within the microfluidic devices. So this is the layout of our homemade Raman optical tweezer. So we have a laser here. This one is a 785 near infrared laser. Besides, we have 2 cameras that can monitor the movement of the bacteria within the microfluidic lab-on-a-chip system. And we have the CCD detector that can generate the spectra. So what we do is, so this is a product founded a few years ago, but still ongoing. We firstly induce the bacteria into the dormancy state. So the reason we want to study the dormancy state of the bacteria is because in the food industry, there are a lot of concerns regarding dormancy state of the bacteria, such as the one that we call the viable but non-culturable. Because in the food industry, it's still very challenging to identify the viable but non-culturable state of the bacteria, which is one type of the dormancy state. So in a bacteria population, there's always a kind of a mixture, including the healthy cells, the injured cells, the dead cells, as well as the dormant cells, which is the viable but non-culturable, the VBNC state of the cells. So such a bacteria population can be introduced into the microfluidic lab-on-a-chip system that we designed and auto manufactured. So after that, we used the Raman optical tweezer to trap single bacteria cell. And in the meanwhile, we can immediately collect a Raman spectra. On the basis of the Raman spectra, as well as the established database, we can justify if this cell is a healthy cell or a dead cell or injured cell or the VBNC bacterial cell within a few seconds. So once we identify this single cell is belong to the dormancy state of the bacteria, which the VBNC cells, then we can manipulate the cell to move from the channel of the microfluidic system into one chamber. So this chamber later on will be harvested for future characterization on the basis of the single and the pure VBNC bacterial cells, for example, the single RNA sequencing. So this can offer a lot of more tools and also offer a more accurate characterization of the bacteria on the basis of the pure bacterial population. Because conventionally, even though you claim you harvest the VBNC bacterial cells or any other group of the bacteria, actually it's not 100% pure. There's always some impurity from other subpopulation due to the nature of the bacteria mixture. So the first paper just published this year in Analytical Chemistry. So in this paper, we developed a new machine learning algorithm. This is we call the spectral recovery condition generative adversarial network. In short, it's called the SERGN. So the SERGN, in short, can tremendously increase the resolution of the spectra and reduce the noise that we collected from the raw spectra. Due to the time, I don't go to the details, but you can see this is the schematic illustration, how we use the Raman optical tweezer coupled with the microfluidic lab-on-a-chip system for the capture and the characterization of single bacterial cells. Because each spectral characterization can be finished within 1 second, then the noise is selectively high. So, but by coupled with the SERGN, we can tremendously increase the resolution of the spectral as well as reduce the spectral noise. So in this case, this can offer a more accurate prediction about the species of the bacteria that we collected, coupled with the Raman optical tweezer and the microfluidic lab-on-chip system. So for the works that I have presented so far, those are based on the Raman spectrometer that we called benchtop Raman spectrometer. It's pretty bulky, it's pretty expensive, but it's very accurate. However, there are a lot of applications that use either the portable or the benchtop, the portable or the handheld Raman spectrometer to achieve the in-field or on-site detection. So this gives us the motivation to develop a homemade portable Raman spectrometer. So this is a spectrometer that we developed. So you can see the size is comparable to the mouse. and the weight is about 2 kilograms. So you can carry the portable Raman spectrometer device to go to the grocery store or go to the farm for different applications. So there are 3 layers of the control panels for this homemade portable Raman spectrometer. The bottom layer is to control the performance of the laser. So the laser here we used is a 785 near infrared laser. So the middle layer of the control panel is to control the performance of the spectrometer. And the top layer of the control panel is to share the collected Raman spectral via the iCloud system. And eventually the received or collected Raman spectral can be immediately transmitted to the PC in the central lab for further analysis, such as chemometrics, multivariate statistical analysis, or even machine learning. So by using the portable Raman spectrometer, we do a lot of different applications, such as food authentication study to identify the species of the fish on the basis of the chemical composition. So conventionally, we need to put the fish sample back to the central lab, use the benchtop and the very bulky Raman spectrometer to correct the Raman spectral feature on the basis of different chemical composition of each type of the fish. But right now, we can use the portable Raman spectrometer to do the spectral collection in the grocery store, even with the packaging system. And then the collected Raman data can be immediately shared through the iCloud system back to the central PC in the lab for further analysis. So this saves a lot of time and the labor for food authentication study. So I want to go to another area about using Raman spectroscopy to do characterization of microbiology or bacteria. So this is what we call the surface enhanced Raman spectroscopy. So for all the previous sections that I mentioned, this is what we call the normal Raman spectroscopy. So the major disadvantage for normal Raman spectroscopy is the relatively low sensitivity. This is due to the nature of the Raman scattering, because every 10 to the 6 or every 10 to the 7 photons from the incident laser can only generate 1 Raman scattering photon. So this is the nature that why Raman scattering signal is very weak. So this gives a problem that how we can use the conventional Raman for trace level detection of very low level of the chemical or microbiological hazards, especially in a complicated sample matrices such as food. So the solution is to couple with the nanotechnology, either use a gold or a silver nanoparticles or nanosubstrate to tremendously enhance the very weak Raman scattering signals. So the principle is that by couple with the silver or gold nanoparticles, there is a huge increase of the localized surface plasma resonance due to the electronical field that generated for the interaction between the incident Raman laser and the gold or silver nanoparticles. And this interaction will significantly enhance the very weak Raman signal intensity, and theoretically, by using the surface enhanced Raman spectroscopy, it is able to detect a single bacteria cell or a very trace level of the chemical contaminant in the sample matrices. So the key to apply the SIRS technology is to fabricate the very good SIRS substrate in order to increase the performance of the sensitivity of the Raman spectroscopy technology. So in my research group, we develop different type of the SIRS substrate in order to enhance the very weak Raman signals derived from the samples. So such as a leafy structure. So you can see the leafy structure is at between the nano and the micro size. So once the analyte can fall onto the surface of those leafy gold structures, then a tremendously enhanced Raman signal will be received. So this is another example. We designed this a hollow structure on the basis of the silver silver nanostructures. So once your bacteria or viruses or even chemicals can fall into the hollow structure, then there's a huge enhancement of the electromagnetic field within such a space. Therefore, the Raman scattering signals can be enhanced. But all in all, what we want to make sure is your analyte, no matter the bacteria, the viruses, or the chemicals, will fall between the interface of two individual gold or silver nanoparticles, because this is the one that we call the hotspot. So the hotspot is the key part to enhance the very weak Raman scattering signals. So I introduced the surface enhanced Raman, but we have another challenge. The challenge is that when we use the source to analyze or detect the analyte within a very complicated sample matrices, especially food, because we know in the food, there are a lot of components, there are a lot of macromolecules such as proteins, lipid, polysaccharide, nucleic acids, etc., etc. All of those macromolecules within the food matrices will unfortunately contribute to the Raman scattering signals. So there's highly likely that those contribution of the Raman spectral features from the food residues, from the food components, will mask the Raman or the search signals from the targeted analyte. So therefore, the key challenge of using the search to detect trace level chemical or microbiological hazards in food is how we can completely or maximally eliminate the interference of the food components. So therefore, separation element is the key that can couple with the search to achieve the successful detection. So many of you heard about aptamer, heard about the conventional antibodies. So those have been applied and coupled with the search for such application. But here I want to introduce another group of the separation elements that we successfully conjugate with the search. This is called molecularly imprinted polymer. And in many publications, they also call the molecularly imprinted polymer as the artificial antibody, or they also call the plastic antibody. So the general principle is we need to synthesize such a polymer. So let's say we want to detect a chemical A from a food matrices. Then we use the chemical A as the targeted molecule, or we call the template, to initiate this synthesis of the polymers. So the first step is we select a certain functional monomers that can have a self-assembly with the template molecule to design such a complex. So after this complex has been formed or synthesized, we will introduce the cross-linking agent to initiate the polymerization process. And eventually, such a polymer will be synthesized. But keep in mind, your template molecules are still inside of the polymers. Then we need to wash off the template by using certain type of the organic solvents. So after the template is successfully washed off, then your polymers is ready, because now your molecularly imprinted polymers will have the complementary structure to specifically and smartly recognize the shape and capture this template later on once the polymers identify the template in a complicated matrices such as food. So in this way, we can use the molecularly imprinted polymers to smartly identify and capture the targeted template molecules from a complicated sample matrices. So we use this idea to develop a lot of MIP-based surface-enhanced Raman spectroscopy sensor for identification or detection of different type of the targeted molecules. So in this study, we target the detection of melamine in the dairy infant formula powder because melamine is one of the representative adulterant. This molecule has a very high level of the nitrogen compound, and the people artificially add the melamine into the dairy product to mimic a relatively high protein content. So because our targeted analyte for the detection is melamine, and then we use the melamine as the template to synthesize these molecularly imprinted polymers. So once the MIP is successfully synthesized, we add it into the solid phase extraction cartridge as the SPE for the separation of the melamine from the dairy products such as the whole milk or the dry infant formula powder. And all the milk components will be removed, and only the melamine residues will be enriched and separated. And then we add those enriched and separated melamine compound onto the search substrate. Followed by that, we can collect the search spectra for the accurate detection of the melamine in different type of the dairy products. So I have two more slides to give you some further application, how we use the microfluidic lab-on-a-chip coupled with the surface-enhanced Raman for microbiology application. So the first application is to use the microfluidic coupled with the SIRS to detect the antimicrobial resistant bacteria. So this is the design for the microfluidic device. So basically, we generate a lot of mineral oil droplet, and in each individual mineral oil droplet, there include bacteria suspension, include the gold silver nanoparticles to achieve the SIRS application. Then we shoot the laser into a specific location within such a microfluidic device. And along with the movement of each individual mineral oil droplet, then we can collect each individual SIRS spectra for different biological sample trapped inside of each individual mineral oil droplet in order to achieve a high throughput screening of the spectral collection for hundreds or even thousands of clinical bacteria samples within an hour. So compare with the conventional multi-local sequence typing for bacteria typing, or by using the conventional PCR for antimicrobial resistant gene characterization, we can use a optofluidic SIRS platform to achieve both the typing and the detection simultaneously, plus the time to finish such characterization is tremendously shorter compared to the bacteria typing or the PCR methods. This offers an alternative to the clinical characterization of antimicrobial resistance bacteria. So the last application I want to introduce today during the presentation is that we, for the first time, couple the microfluidic lab-on-a-chip system and the Raman spectroscopy to develop such a platform for the cultivation and the characterization of the bacterial biofilms. So the biofilms can be cultivated within a microfluidic devices, followed by the characterization of the chemical composition of the bacterial biofilm. So here you can see during the cultivation from 24 hours up to 72 hours, the bacterial biofilm continuously grow and develop within the microfluidic devices, and all the peaks characterized here are related to some chemical composition of the bacterial biofilm, especially the extracellular polymeric substance, in short, the EPS. So conventionally, you need to do the dyeing and followed by using the confocal laser scanning microscope for the characterization of only the thickness of the biofilm. But for now, we can use the Raman coupled with the microfluidic devices to not only characterize the thickness, but also the chemical composition of the biofilm at different maturation stage. And also, by using the partial least square regression model, we can find a very good correlation between the conventional confocal laser scanning microscopy data for the biofilm thickness, as well as the Raman scattering signals. So within that, I will wrap up my presentation today and open for any question. Thank you.